So we'll go through some of the basic concepts of immunology tests, and today's lab is based upon antigen antibody reactions, and chapter 6 will talk about most of the basis of the tests that we normally have in clinical practice. And I'm going to take you through, because that is the only time that we'll have you learn this and take a quiz at the end for chapter 6, plus the blood lab. And this chapter will not be included in the upcoming exam. So we'll talk about some of the basic antigen antibody reactions. And as you can see over here, immunoassays, cellular assays, and cell culture, these are some of the common tests that we employ these days. And I'll also talk about, if I do get time, for the experimental animal models that we have today, and some of the things that are coming as a newer generation of tests in terms of gene expression. How many of you, I think this was this morning's BBC News, that they got permission from the government to edit the genes. There was one big news from England that they gave this, they have this fertilized ovum, and they want to play around with some of the genes. So you can see they will go and kind of edit those genes. If some of the genes that they found out are leading to cancer or some of other problems, they want to edit that. So things have gone that far. They're talking about gene editing. So that's going to be a very interesting thing. And if you remember, when I came to talk in Dr. Petrova's class as well, I was saying that it's not only pharmacy school, it's all the schools, including medical schools, are going through a big change of pace, curriculum revision. And at this place, we are going to adopt a new, we call CAPE outcomes by 2016. So you see a lot of changes, but I can give you a little bit of update on that. Things are moving towards a direction that we professors hate to see, because then we have to learn and teach. The medics who are, out in their practice, they want to go and re-educate themselves. There's going to be a challenge for everyone. Right? It's going to be a challenge for everyone. And since you are also going to be in a competitive market, so it's better for you to at least have a little bit of understanding or grasp over that if you want to remain competitive. Okay? So gene expression. Okay. Now, it goes without saying that for the basis of the test is that we are looking for a very specific, I don't remember how many times would I have told you that these antigens are specific and antibodies are specific. So this means if you want to do a test, there's going to be a very specific test. Okay, so if you tell me what the antigen is, I will make an antibody to make sure that that antibody locks with that antigen. So for example, all different hormones, for example, or all different bacterial infection, viral infections, they will have an antigen. So we can take that antigen and make antibodies and make them react in a culture plate, in a tissue culture plate, on bench top, and do a test. That's what it is. So most of them are antigen antibody reactions, and they are the basis. And again, is a very exquisite specificity, very, very exquisite specificity. But still, there are some kind of technical problems that we'll go through, and I'll let you know what they are. One of the common things that we normally do is they say serotyping a microbial AG by anti-sera. For example, you must have heard, hey, E. coli has come up and they have figured out from spinach, from such and such thing, there's an E. coli outbreak. They want to match it up with the production plant in Atlanta or Florida, because 2 different patients were reported. Right? Now you have to think and trace the history of that particular E. coli, which originated from a plant, and that bacteria sat on that spinach being delivered in Florida and delivered over here in Chicago, and somebody consumed it. They want to find out, are they the same? So what they will do is that they look at this microbial antigen which may be circulating in the blood of the patient and match it. So they will give that a very specific number, say 0, 5, 7, whatever number they give. Like you must have heard N1H1, N2H2. These are the flu types that we see. So we give them a identification number. And how do we do that? That again is the basic, we call serotyping. Serotyping means we test your serum and type your antibody. That's what pretty much it means. Now in real time, the interaction of antigen antibody that we've been talking about over time, and you remember that mnemonic I told you for pain, precipitation, agglutination, and immobilization and neutralization. And then also activation of complement as well, because we can add some of the players to the antibody structure. So this will remain as some of our chemical reactions that will tell us whether the test is going to be positive or negative. Okay? Okay, let's go to the figure. And I think you'll agree with me that, and I myself can tell you my struggle when we used to read these notes many, many years ago. We would rather draw our own figures and all that stuff, cartoons to remember it. Because visual, most of us are visual learners. It's difficult for us to kind of comprehend if there is no figurative thing over here. So it makes your job easy as well. Now, 1, A, B, C, D, E. So what you are looking at over here is that you pretty much know the structure of an antigen. You also know the structure of an antibody. You also know 2 parts of antibody. Right? You know a heavy chain, then you have light chain. Then you know FC portion, then you know fragment for antigen binding. Right? And if you remember one of the slides, I said 2 enzymes can chop those antibodies at different places. If you chop them up in a way, you may end up with one structure having 2 hands. Right? Or if you chop them a little bit higher, so you may have 2 different fragments split together. They all have those capability, and we use those capability in enzymatic digestions. That's what the whole point for these immunological testings are. Okay? But also keep in mind the protein binding sites, the antigen binding sites are pretty much stable. We are not chopping them off. We are not. Okay? Now let's see, for example, test number A. This line is an antigen hapten, antibody fragment and complex is formed. So the whole idea is, as a given example, that you have an infection in your blood and there is an antigen of that particular bacteria. And we will take your blood out, serum out. This serum is like a clear portion of the blood, not the cellular one. Cell-free part of the blood is called serum, right? Or plasma. So there you will look for proteins, plasma proteins. Okay. Now, this example pertains to today's lab as well. You will see those particles over here, which are called antigens, or red-colored structures, and you will see only one antigen on all of them. Right. So if you remember, if there was only one antigen on the top of surface of any structure, let it be your red blood cell, which you are going to do today, or any other tissue. So we call it a univalent. There's only one part of that antigen that is going to be exposed to an antibody. Okay? Compare that with a multivalent, where you have 3 different structures. Make sense? And you also will see on the top over here, if you were to allow antibodies to go and act with them, for example, let's say these are red blood cells. Okay? So this red blood cell have a polysaccharide antigen expressed on top of that. Like for example, you must have heard that, okay, I am blood group A, for example. I'm not though. But so this means I have an antigen for A. So you see over here, there's an antigen for A. If I want to find out if any one of you has the same antigen, so I will give you an antibody for A. So you will have a anti-A antibody. Okay, so they will fix together, and then they will form a complex. Right? They form a complex, and you will see today on your plates today that complex is visible to the naked eye most of the time. You'll see it for yourself. I mean, you can use it with a microscope, but you're not going to see antibody because the size is very small. But you can see that in this case, one antibody can bind two antigen expressed on two different surfaces because of the nature of the antibody. Since these cells are floating, so it's just gonna, I can grab two people with my hands, right? But I cannot grab this desk over here and that bench over there because I cannot stretch myself. So antibodies have this stretching capability. But if things are floating, running, so I can grab two. That's what these antibodies did. Did you notice that? Okay? But the other thing you will see is that they are forming small complexes. Small complexes, because the antibodies per se are not joining with each other. So if you look at them, it's difficult to see because they're very small clumps. If you compare this one, for example, it has three different antigens. So you pretty much, again, give them an anti-A. But in this case, what has happened is that one red cell can possibly bind three different antibodies. Okay. Now you wonder why is it that it can bind three antibodies. So you keep in mind, for example, if this was an exposed area, right, and this is in Fab portion, so if two antibodies go for the Fab portion, right, the third one is available for the third antibody because it's on the other side. So there's a physical kind of discrepancy between binding sites. That's the example I say that IgM has valency 10, but at a time it can only bind to a lesser number of antigens because if you use 3 of your arms, the other 5 arms cannot be used. Does that make sense? So you will see over here A anti-A cross-linked complexes. So that's what we call complexes, but they are cross-linked between each other, and you will see them more obvious under microscope. Okay? That makes sense? Now, we want to make it more specific, or we want to save time, we want to save energy. So what we can do is that we say to ourselves, you know, what's the point of using too much of an antibody, because by the end of the day, only these portions are attached to this antigen, and these are redundant, isn't it? So let's cut them off. We kind of improve the technique. So what we do is that we will chop it off, because we know FC portion is not involved over here, we already know, right? We know that the antigen is going to bind with the antigen binding fragment. So we will, instead of using anti-A antibody, we use anti-A fragment for antigen binding, right? Because there's no need for FC. You want to improve the test. Okay? Again, you will see over here the anti-A Fab complexes are formed, but the problem over here is that we missed the point that the antibodies would not be cross-linked with each other because they are not sharing anything. You just had to cut off one of the arms and just stick it over there. So again, they are not cross-linked. Now what we do is that we say, okay, let's try having two fabs, you fab 2, and try it again. This time you see they are cross-linked complexes. Okay. And still we thought, what was the point of FC? So we pretty much improved the assay. We want to save time. We just want to use 2 fragments of fab and have them attached to the antigen and also have them cross-react with each other so we can see the cross-reactivity. Okay? But remember over here, in A, B, C, and even D, the antigen was the same. So we just employed anti-A antibody, isn't it? But the question is, not all of you will have A blood group. So you can have A, B, AB, or none, O. So these are based upon ABO antigen classifications. I'm classifying you on the basis of antigen A and antigen B. So let's say there's another antigen which has ABC. So once we know there are 3 different antigens, so now we have to give 3 different antibodies. You have to give anti-A, you have to give anti-B, and we have to give anti-C, and they basically will bind to their respective antigen because they are specific. Okay? That makes sense? It's pretty much the same thing, but I showed you in a larger picture in the previous one. Now, you also keep in mind that this kind of interaction that we have between an antigen and antibody is non-covalent. That's what somebody asked me a question this morning as well about the first signal where the MHC has this bunch of peptide attached to it and CD4 attaches. So this kind of attachment is non-covalent. So in this case, again, is a non-covalent bond between an antibody and an epitope. So epitope is that particular part of an antigen that really binds to. It's like there's a, what do you call, a lock, but the key has to go to that smaller portion. So big lock, smaller portion. So that smaller portion where you snugly fit your key in is an epitope. And the bigger structure is like antigen. These are relatively weak binding forces, good for us. We don't go and kind of stick forever, right? Otherwise we have a problem. Because in antigen antibody action, we just want signaling pathway. Just like I turn on something on and off and my hand gets stuck to it. I don't want to do that. I just want to do on and off and then move my hand. Turn on the signal, let the signaling go, and move away the inciting or provocating or stimulating chemical. Okay? Now, it's a low energy dissociated by pH, high salt, and cyanates. So these are some of the things that we would want to use if there is such a complex in terms of making those solutions. Because, you know, a low pH, so you can play around with pH, you can play around with the concentration of salt, break it apart. But if you really want to look at the K association constant, so there are very defined way of doing that, right? Just you need to know that they are there where we want to see the intensity of reaction as well. It's not only reaction to be positive. Right? It's just like this. There are some pharmacy schools that just have pass and fail, right? But their passing fail maybe is 90% or something like that, or 80%. But we don't have pass and fail, we have a grading system. We want to see how good your grade is. So we want to see the grade of whatever reaction is taking place. We want to grade it up, high, low, whatever. Okay? So there are constant and equilibrium. That's where it gets very complicated, right? So that's part of advanced biochemistry. We don't want to go over there. The only thing that you need to know is that we have to set up the controls and equilibrium and standards and all that kind of a thing. Okay? For most part of our experiment. The other thing, remember today I was talking about that the affinity is that if your cells find some of the cells showing a weaker affinity for your self antigen, they will let it go. They don't really care. Say, you know, if he's going to come in contact, they're going to initiate the signaling. It's very weak bonding. So that is affinity, as compared to a avidity, right? It's an avid. So they say he's an avid part. That's what we use in English as avid participant or avid sportsman or whatever that is. So this means more than normal, very strong kind of thing. But if you look at affinity, we're talking about the attraction between an antibody or a epitope. Or sometimes remember when the epitope is not strongly antigenic, we add a hapten to it to make it bigger. I will give you an example for the basketball thing. You know, that depends upon the size as well, size of your fingers. When we say avidity, we again are talking about the antibody, but it is univalent ligand. So that's mostly it is for the hapten as well. And we see that more in IgM as compared to IgG because of the nature, the size of the particle. So we will use an IgM when the need be, when you need a bigger structure as compared to where you need strength. Okay, so that again is there for affinity in terms of intrinsic association constant. Okay. Let me go to the figure. Again, you can read it up on your own. Okay. Now, so you have a red blood cell, right? And most of the red cells are unidentified. How many of you do know your blood groups? Okay. Good. But today's test is basically not diagnostic, it's academic. So don't take it as your kind of a diagnostic test, you know, because it's just meant for academic exercise. If you don't know it, and if you get to know today, may or may not be, right? You better get it checked by somewhere else, right? But most of the time, I've seen last time it 100% worked in the last week. So that's good. So we will have a red blood cell, and we will give you antibodies. We're going to give you an antibody for A, antibody for B. And there's one more thing we'll give to you, which is called rhesus. There's another antigen. It's called rhesus. That's why we say, my group is A positive. So positive is rhesus, or A negative. So that's another antigen that has a very strong transfusion reaction. There are tons of antigens. These are not the only antigens, but we are just worried about those antigens that give reaction. Right? So if you mismatch transfusion on AB level, you will have a reaction. And if you mismatch on rhesus level, again a reaction. We don't want to do that. So we'll give you another antibody for the rhesus. That's what we call anti-D. And it is called positive. You say A positive, B positive, AB positive, or whatever you have. So we will give you this antibody. You will attach this antibody over here, and then you will see a reaction. You know, but sometimes what we do is that, let's say in this case, we find out that these antibodies are very small, and they are attached to the cell, but something is missing that you want to do in this chemical test. What is missing in this chemical test? The one we explained to you earlier. If you were to see under the microscope, you will still see cells separated from each other. There is no cross-reactivity. There is no clumping, no agglutination. We are looking for agglutination. That is the basis of today's test. Does that make sense? So what we will do is, we will give another antibody to this antibody, anti-Ig. So what it will do is that it will bind to different antibodies and pull them together. That makes sense? So there's an antigen attached to an antibody, but antibody itself being a protein can also provoke an antibody to an antibody, and then so on and so forth. So you can have 10 antibody to another antibody, because of the nature of that particular epitope expressed over. They're all amino acids. Okay? So this one basically, this principle is the basis of Coombs test. And I did post the videos for you, like 4 or 5 videos for you to at least go passive, Coombs test. So you may want to review that because we at least have one person from each video that we posted. Okay. And you can use your machines, or you can use machines over there in the middle row. But this is basically we call agglutination reaction. Now, I will not go in detail. I would rather want you to, because I think it's better for you to go through the video. It's better explained than what I'm going to explain to you about active and passive agglutination. But the principle is there. And the point I want to tell you over here is that the passive agglutination is very important in terms of clinical application, right? Because the thing is, in this case, what we are doing is that we have antibodies fixed to those cards. They're going to give you Eldon card. These are small cards. So antibodies are already labeled. And you're going to pour antigen on top of it. So you have a soluble antigen, right? So this kind of activity we call passive agglutination. And that basically is very important in order for you to determine your blood groups. This you will do today. Immunohemolytic diseases, I just told you today that some of the drugs will attach to your red blood cell and present as self-antigen, non-self antigen, and your antibodies will attack them and kill your cell. They were your self-antigen, but the way those drugs broke apart, like penicillin, so it kind of mismatched the whole sequence. And that's what normally happens. Then we have all the tests available for rheumatoid and even tests for pregnancy or syphilis. And I gave you an example for, let's say, pregnancy test that again is based upon a hormone that is released in pregnancy. Right? Does anybody know what that hormone is? Huh? HCG. So HCG is released over there, okay? And then you have an antibody to HCG, so you cause that reaction to find out whether it's positive or negative, right? But again, if things get messed up, you may have a false positive. For example, a person can do a random test, no history, no kind of planned pregnancy, and you come out to be positive. Doesn't make sense. Or you can see the person is there in the second trimester and the test becomes negative. Right? So then we will start thinking, troubleshooting, there's something wrong with SCG, something going on. And we sometimes also look at the increasing titer or titer. It should go up. So one time, so these are some of the things that we normally do for passive agglutination, and then do for HIV, we do for syphilis, all these things. Because if you are infected, you have an antigen, in the test we provide you with antibodies. We already have prepared antibodies. Okay, so that reaction. So you can see over here, passive agglutination is one of the commonest tests that we normally do. Okay, then for the precipitation reaction, and you can also see agglutination will mean that in a solution things agglutinate, right? But in precipitation, the whole chemical thing kind of fall to the bottom because of the size. So that's like basic chemistry, right? And then again, we have at least 1, 2, 3, 4, 5, 6 different tests. And again, I would refer you to some of those videos which was better explained than what I could possibly do. That they can be done in a liquid, they can be done in a gel, and they can also be done in electrophoresis, where you kind of run cathode and anode in opposite direction based upon the charge. So there are many ways that you can play around to find out that reaction which is there. Okay? The other important thing that you will see is that when we look at like antigen antibody complexes, we want to make sure that you have a proper concentration. That's why we do the standard curves. Because, for example, if you have over here zone of antibody excess, more antibody than the antigen, then there is no complex over here. So you see very small complex. Or you will see more antigen than antibody. So there's no precipitation reaction that you can visually see. So you have to draw that in dilution. So what they would do, for example, is I'm going to take like 100 microliter of your sample and I'm going to dilute it like 1 in 10, 1 in 100, 1 in 1000 and see which place will be the best match for the antigen, because I don't know how much antigen you have. So I have to draw it on a Packard curve, right, a structured curve, where we will see the best reaction when antigen added is equal to the antibody who had equivalence zone. So you will see these kind of, in the test tube, you can basically see that precipitation. Also in today's experiment, I can describe you, and I think what I want you to make sure that you can go to the eldencar.com. Also, they will also give you on this computer, if you go on the network, on your own network, the whole procedure. We tried this for this year. We wanted to save kind of wastage of paper, so it's already there. So one of the things that I would want you to do is that because I've seen for some of the people it was a big mess, big bloody mess, so to speak, that they were kind of pricking themselves or unable to draw blood, and I was hoping myself if that's what going to create that emergency for the patient. Next time the patient is not going to come to you. It has to be a very clean thing. But again, how many of you have had your fingertips pricked? Your own, by yourself. Okay, that's the challenge. That's the challenge, because you can be kind on other rougher, and when it comes on you and you just put the plunger this far, and that's not going to work. The plunger is very small. So you make sure that you really press it before you push it. Okay. So if you're kind on yourself, you don't have extra pair of plungers to give it to you. So make sure you be bold, right? Is that correct? Okay. Anyway, so you will see that the protocol will tell you that you pour a small amount of water and blood, but sometimes if you have more of a water and less of a blood, then that ratio will be large. You will not see the precipitation. So I would want you to pay attention to the video and make sure that you don't overdo that. And then another thing is there's going to be 4 different circles for you to put 1 drop of blood in each one of them. So you have to be quick. You don't want to let it dry and you kind of taking 15 minutes. By the time you come back, it's already gone. So make sure that you give a good prick, let the blood come out, and I can assure you that you have capability of even donating half a liter of blood. So one drop is, I would say, 2 microliters. That's not a big deal. You're not going to have anemia because of that. But you can have vasovagal shock though. Let us know if that is the case. Everybody would know though if somebody has that. Okay. Again, I don't want to go in detail. It's even unnecessary. But we can also put antigen and antibody on an agar plate, because the thing is this, because some of the microbial like bacteria will grow on agar plate. So they will grow and we can put antibody. So we have more than one ways of using the same technology of antigen antibody. But you can see from here, if you have an antibody and antigen, they will move towards each other. So that's kind of an affinity that they have in terms of movement that is here in some of the gels. But anyway, so this is, if the antigens and antibody basically see each other and they will have a identification. For example, say that this kind of thing will show the pattern of identity. So we'll see for B, it's a pattern of non-identification because they're moving in opposite direction, right? As compared to identification. But the point over here, regardless of going in detail, is that antigen antibody are very specific and we can put them on a gel and they will move towards each other. And many times we do that to save concentration. We can also do on a different radial diffusion as well. So you can again put different diffusions of a known concentration of antigen. You can have like A, B, C, D, known concentration of antigen, and then put antibody and see where is the biggest complex. And you can pretty much see from here if you want to put on a graph as well. looking at this distance, as compared to this distance, you can pretty much determine. Again, we call it the radial diffusion assays. A little bit complicated. The only thing that you need to know is that depending upon the concentration of antigen, because one of the parameters is fixed. You can change the concentration of antigen and have a fixed antibody. That's where you want to make a chart. And then you can come out what's the best fit in this case in terms of titration, concentration titration. And you wonder, why do we need that? I remember one of the P2 students was telling me that they were asking whether that student was vaccinated for MMR, and she doesn't remember that. So they wanted to look at the titer of antibodies in the blood. So we can do that. Okay. So that's where we want to do in terms of those of antigen and antibody. Okay. This was, again, I told you the third thing, immunoelectrophoresis. We talk about that. You know, depending upon the size, the heavier will settle down, lighter will move up. But we can separate. You can see if you learn a pattern immunoelectrophoresis of serum proteins, heavier will stay because they're moving in this direction, transfer IgM, IgG, IgA, and IgG. You pretty much know that they will settle on that electrophoresis apparatus based upon this size. Okay? All right. Also keep in mind, these are the samples that you usually get are serum samples. We call serology because we find antigens and antibody in serum. So we're looking at serum samples. Now this test basically is give you a Western blot. We call it Western blot. And if you pay attention over here is that there are two infected, HIV infected patients over here. Okay, these two. And then this is a control. So what we have is that we already know the causative agent of HIV. So we have taken those specific antigens from this virus, from the, for example, core, from the core of that virus, then the other protein, and one of the enzyme over there, some other protein, some envelope protein, like 4 or 5 different antigens we have picked up. We made antibodies, 5 different specific antibodies, and now we're going to run a test. So what we'll do is that we'll run the sample of those AIDS patients, right, in a gel, and add all those antibodies with the control as well, which we know is negative. So you will see over here, once the antigen and antibody react, it forms a band. So if you, in control, you don't see any band because the control is not infected with the HIV. But the person who has HIV has those antigens. So if you use these antibodies over here, positive, 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 but you can also see, why do you run more than one? Because different patient may be positive. This one is a classical reverse transcriptase. This is a classical enzyme for HIV. So it's positive in both. So we know that it's there. Okay. The other thing what we normally do is that we also look at viral load. So the HIV patient's blood is teeming with viruses. His secretions are teeming with viruses. Saliva, right? All these body secretions, and you are giving the patient the therapy. So we want to see if that therapy is working. The target is you want to bring the viral or antigen load down. So we will also test them and find out if the load is going up or down. So that again is a test for what do you call HIV. Or there's another spin to this one is what we can do is that we can also have antigens over here on this gel and take patient's serum because he would have antibodies as well. Because all those antigens will invoke an antibody response. So either we can test for antigen or we can test for antibody. And by both cases, they would react with each other and give us a positive reaction in terms of HIV. Okay? So we'll discuss that in detail when we talk of virology. All right. The other type of test that we normally do is, now we're going to become a little bit more kind of technical, because it's just like pregnancy test, positive, negative, no big deal, right? But when we want to do, for example, your thyroid hormone level, right? So no, we don't want to see positive, negative, because it's positive anyway. Thyroid should be there. We want to quantitate that. Okay, so you want to see exactly what is the unit of that. If you want to do that, so what we do is that we label the antigen. We label the antigen. So we label the antigen so that we can count those labels. And many of them are radioactive. So they're going to go through the scanner and you say 50, 60, 70. In this case, you can see we have 9 units of labeled antigen, and we have an antibody, and then they kind of attach to that, and we can look at the structure bound versus unbound. So we can run as a control. So we can do another improvement to that test by adding a label to an antigen. Okay. But in this case, what we can do is that we can play around which one you want to make a variable. If this is variable, you want to make this constant, so that we pretty much know, because you're testing variable towards a constant, create this standard. Okay. All right. Now in this case, we call it radio immunoassay. Again, you can have both labeled versus unlabeled, and pretty much the same thing that you can differentiate and figure out exact doses, because you already would know already what is there in terms of labeling. So you have a constant, and you run on the machine, and you find it out. We do that many times for thyroid hormone, other hormone assays, radio immunoassays. That's what we say. Okay. Now, this is, I'm pretty sure that there is a good video on there, but this is important. And many a time, this is a first test that we normally do before going to second test. It's called ELISA. It's called ELISA. So it's not ELISA. Somebody's name is there. So we call enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. But understand the principle behind it. The principle is that we have this plate. So how many of you have seen 96 well plate? This small plate. I think we showed you the 96 well plate. So it has 96 wells, so you can test 96 samples at the same time. It's very good. So what happens is that in one of the well, we coat it with antigen. Remember I said either you can, if you have antigen, we look for antibody. If you have antibody, we look for antigen, either. So we coat it to the plastic well and then block the sites, because this is based upon binding. You want to block any site which you don't want to, because if you don't do that, you may have false positives, because it's non-specific binding. Okay? Now, what we do is that we block this protein over here, so the antibody would not bind to these empty slots. Then we have an enzyme labeled antibody. So we have an antibody, we label it with an enzyme. That's okay. Enzyme linked. That's what ELISA is saying. Then we add this on top of that plate, right? And it should bind to these ones, because it has these binding things, right? And then we add a substrate to that enzyme reaction that will give us a color. And we look at the color and then do the analysis for that. So that's the basis of enzyme linked immunosorbent assay. Okay, and that's the test that we normally first do when we do for HIV before we go for Western blot. Okay. Then, so far I've been talking about antigen, antibody, but again, remember that there are cells as well, because humoral immunity, antibody, antigen, and there is cellular for CMI, cellular immunology, cell-based. So we want to look at cells as well. So how do we look at cells? So what we do is that because cells will have antigen expressed on the surface of those cells, so we want to tag those cells with antibody, fluorescent antibody. That's what we normally do. Fluorescent antibody, you can see over here, fluorescent antibody, right? So SLE or whatever test we're doing, and we call it a FACS analysis, right? It's based upon activated fluorescence to look to the machine. It's there in biology. FACS analysis. So what happens is that it has a small little a tube, and the cells will come and then pass through that bar. Again, I think I saw that a good video that you have posted to you. And the laser beam of different colors will hit it. So you can have like red, blue, green. It will hit depending upon that specific antibody that you label it with. And cell drop one by one. So it will not only count, label, even sort as well. If I was to take your blood and say, I want to take all your CD4 cells separately, the machine will do it for me, based upon the antibody. So that's called fluorescence. There's an indirect or direct, depending upon whether you have a primary antibody or you don't have a primary antibody, but pretty much the principle is the same. Okay, fluorescence-activated cell sorting analysis, that was cell, and that's the common method for us to look at those CD markers. I told you CD4, CD8, and all these things. And CD is acronym we already talked about. And I also told you that the CD number comes from the International Union for Immunological Societies, because it follows a rule: CD4, CD8, CD26, and there are hundreds of them. Each one of them has a specific assigned number. Okay, now this is the principle. Cell mixture is there, you have fluorescent antibody, you tag them, pass through that machine, there's a laser beam, it's going to fall over there, there's a detector over there, it will sort things out or label things out. Pretty simple, very specific. So all those, even development on top of that is that now we have some of the antibodies that can be permeabilized, they can go into the cell. And even scan those kinases and MAP kinases or ZAP70 or whatever we're talking about, LAT, calcineurin, because antibodies can go, we call permeabilization. So permeabilize, and then we go over there. So that's what it is important for. Okay. There are some other assays, immunoabsorption, I don't want you to, fuss much about that. But the important things are other cellular assays. Remember I told you what are the functions of T cells? What are the the effector response of T cells? Proliferation. Huh? Producing cytokines. Killing. Correct, killing. So these are the three things that we can assess whether your T cells are functional as well. That's what we say. So what we're doing, assess excess lymphocyte function. That's what we are doing, right? So we look at proliferation, and we can look at antibody production as well for B cells, whether they are able to do it, and then we look at their killing. So these are very specific tests that we normally do, and we want to make sure that you do that. Okay? So for proliferation, we can see if they are 10 and they become 100, we stimulate them, right? And then we see if they are progressing. And we stimulate antibodies, B cells, see whether they produce antibodies. And we put like cells and these T cells together and look if they kill each other. When they kill each other, we notice that. Right? Because when a cell either dies for inflammation, apoptosis, we have markers for that. So we have markers to figure out whether the cell is dead or not. What is the normal marker, those of you who have worked in lab, for cell death? Simple microscopic marker for cell death. Those of you who have ever worked in a cell culture lab. What is the dye that you want to use to find out if the cell is living or dead? None of you have worked anywhere? No research experience? Trypan blue. You were close, blue. Trypan blue, right? But propydium iodide, annexin, these are the things that we normally use clinically. Okay? And then again, what we can do is that, we have those cells, and then we can develop cell cultures. Remember that if we have CD4, we can develop them. We have CD8, we can develop them, so on and so forth. And then again, I'm pretty sure that you will be interested in that how do we make antibodies commercially. So you can see from here that we already have a mouse that we inject with an antigen, and then we give a cancer cell. So we give like a cancer cell, and we pick up these cells and put the cancer cells with them. We call it fusion, because myeloma cells are like cancer cells, and then they start producing antibodies. Remember when I showed you the slide over here where it was uncontrolled antibody production? I said this is myeloma. So we use that characteristic for those cells to produce antibodies for us. And then again, we will clone them and make specific antibodies. That's what we normally do. But this was a big technology, hybridoma. We call this hybridoma. So this cell over here, you see hybridoma cells, immortal cells, because cancer cells are immortal. So you can produce as many antibodies as you may feel like, okay? So that's commercially produced. Lastly, some of the experiments are done on animal models. We don't want to test on human, of course. So we have developed all those mice. Remember I told you nude mice. What was the nude mice I said in the class? The mice that doesn't wear clothes or something. Why was it called nude? It doesn't have hair. Very good. The mice that doesn't have a hair. Why was it called nude? Hmm? What was knocked out? That's called gene knockout mice. That's the nude mice. It's called gene knockout. Why is it called nude? Very good. Thymus was removed. When you remove thymus from the mice, it doesn't have ability to produce T cells. We call it nude mice. That is correct. And actually, you just have to read over here. Many a times, isn't it correct? Many a time I ask the question, the answer is already there. There you go. You can ask people, they will help you with that. Not many of them, most of the time. So whenever I ask the question, the answer is already there. Thymectomice, so remove the thymus, and there's athymic nude mice. Okay? What is a skid mice? I didn't tell you about that, but just those of you who are ahead of me, reading ahead of me. Not the Googlers. Severe combined immunodeficiency. Severe combined immunodeficiency, we call it skid mice. Okay? Now, your friend knockout mice, what we normally do is that we have an ability, and these experiments were done in mice before they started this morning doing on human beings. I told you this morning news. Receptor editing. So we're going to go. How many of you have heard about designer babies? What is that? Correct. Yeah. So what they do is that you go for baby shopping and you say that, I want to have a baby, this color of eyes, this color of hair, this. So they're going to put everything, kind of match this match, and then they would rather look for the sperm from the sperm bank. But that's a crude way. That used to happen before. But now what they would do is they would kind of take your order, right, mix and match, and inject you with that particular sperm, and there you go. It's like receptor editing. That we have this ability to do that. We can insert or take out those genes. And that's crazy, but that's where it is. Okay, so it's more of a control on that. But the problem would be just like there was a time we were talking about cloning. Say you're going to clone Michael Jordan. Okay, you can clone Michael Jordan, but there is no guarantee that he's going to be a good basketball player because skills are different than. Is that the point? So there are still some things that we are working on, but these are possible. Transgenic mice and knockout mice. So we can insert the gene, and many times you wonder why do you need to take the gene out, because that gene is causing cancer. So instead of that experiment we talked about, removal of breast or removal of what you call uterus, this and that, we say, hey, let's remove your gene of that segment, type it up, let's clean it up, right? Right now it's all experimental, but these things were done on animals, poor animals, but that's what it is. I remember I was in, I think 2 years ago, I was in NIH, and the Frank Collins is the chairman of NIH, and there was a big show, and he was there making an announcement that they knocked out the genes and they're going to have this and this, and there comes a woman and she starts shouting, and, oh, you killing animals, the animal rights people were there. Disruption over here. But the problem is that that's the way the science works. I mean, I'm not like anti, but that's the way it is. But if the argument is you killing a life, well, if you're going that far, then mycobacteria and viruses are also life. You better not kill them as well. So that's going to put your production in danger then. If that's the underlying kind of philosophy. But anyway, so there are animals available for you to those experiments. Okay, so I'll stop here.